Hello, I'm Les Bennett, and I'm with the American Association of Pharmaceutical Scientists. It is my pleasure to host this special series of interviews with individuals who are recognized for their innovations and contributions to the pharmaceutical sciences. Speaking on behalf of AAPS, one of our goals is to develop a series of videotapes in which we invite eminent pharmaceutical scientists to discuss their points of view on the events, issues, and challenges that have shaped the direction of the pharmaceutical sciences. We also ask them to give us a little history of how they have participated in this development, and finally, their perspectives on where the pharmaceutical sciences are headed in the future. Today, it is our pleasure to have the opportunity to visit with Dr. Jerome P. Skelly, an eminent pharmaceutical scientist, most known probably for his leadership at the FDA. Jerry, welcome, and glad you're here today.
Thank you, Les. I appreciate it very much.
So I think it'd be good for the audience to you give them a little background of your sort of CV background.
All right. When I started college, I really didn't know what I wanted to be. And I came from a very limited background, and every time I got to a new subject, whatever it was, that was what I wanted to be. And eventually I settled on chemistry, and I got my bachelor's, master's, and PhD at Wayne State University in Detroit. My mentor was a fellow by the name of Albert Boyle, who was a full professor of chemistry and an associate professor of medicine. He was a PhD, MD, and he was interested in quantifying medical phenomena, and we were particularly interested in those days in working on arthritis, and so my PhD was in connective tissue, working on connective tissue using a technique which was brand new in those days called pulse labeling, in which you put in a short-acting radioactive component and traced it going through the tissues. After my PhD, I joined the FDA and gotten very much involved with the biopharmaceutics, with the preliminary to the biopharmaceutics program, and eventually went out to study under you and Malcolm Rillin and Sid Riegelman to pick up pharmacokinetics.
Good. Okay, so give us a little history beyond, because after the FDA, then you did some industry.
Yes. After I retired from the FDA in January of 1993, I became a consultant to Berlex Laboratories. And in those days, this was a company that was fully owned by Schering AG in Germany. And the Germans, being excellent chemists, kept changing things as they would improve their assays. This, however, did not make it to the NDA, and the American company was not able to persuade their German supervisors that they had to do this, so they hired me to help persuade them. We figured this would be a 3-year project. We got it accomplished in 2, and then I joined Copley Pharmaceuticals as the executive VP for research and regulatory affairs. They had some problems with the FDA, but told me they had not done anything wrong. Unfortunately, they pleaded guilty to generic drug fraud, paid a $20 million fine, and I resigned immediately. I set up my own consulting business and consulted for the industry, occasionally teaching at colleges of pharmacy. I took an adjunct professorship at the University of Cincinnati, and retired finally 2 years ago. I figured at age 76 I had done my share.
Okay, great. So Jerry, since you were at the FDA in the beginning of the biopharmaceutics bioequivalence development, I think it would be good to give us a little history back there, what was going on and what were the issues. And you might start with what you thought you were going to do with the FDA when you first came in.
Okay, that's a very interesting thing. The FDA actually got in touch with me because they had several NDAs submitted by a French firm using the pulse labeling technique, and they had no one available to review that information. I was invited to Washington for an interview decided to take the job and came here. I joined the metabolic endocrine division as a chemist. After a month of reviewing this material, I asked a number of questions requiring a considerable amount of extra data. The French firm decided it was not in their interest to do that, so they did not pursue it. After that, there really wasn't anything that I had as an assignment. So my supervisor came in with several Federal Register books and told me I should get acquainted with the regulations. I sat down and started to read them, and I was horrified. I could not understand all the legalese. I was not interested in it. I was interested in science. I went in and told him I was going to leave. However, I would stay until he could hire somebody else. That afternoon, I was in the office of Marion Finkel, who was a physician and the director of the division, and she persuaded me to stay and that I would get some new assignments. My first assignment the following day was on a case of maple syrup urine disease. And this is a case where you have 3 branched chain amino acids, leucine, isoleucine, and valine. And these children have a defect. They are not able to metabolize that branched chain. And so these amino acids build up, cause toxicity, and spill out into the urine. If you smell the diaper, it smells just like maple syrup. This physician in the boondocks of Alabama discovered this child, got to the FDA requesting amino acids, a source of amino acids that did not contain these 3 amino acids. Unfortunately, this went through a bureaucratic procedure, and by the time it got to my desk, the child was moribund. My supervisor told me I should ask for manufacturing controls, which was the bureaucratic way of dealing with this. This is how the chemists, were instructed. I refused to do so. And I told him I wanted to solve the problem. That afternoon, I was back in the office of Marion Finkel, who agreed with me and allied me with a brilliant pediatrician, a gal by the name of Martha Bennett, who was a physician and had very good contacts with the Pediatric Society. And then she aligned both of us up with a fellow by the name of Alan Forbes, who was an MD, and he was in the Bureau of Foods because this was a food item. The 3 of us then began to proceed to try to solve the problem. Alan worked with the bureaucracy inside, Martha worked with the Pediatric Society, and I worked with a group called Cal Farm in California, who was willing to go ahead and get purified amino acids, because in those days there really weren't any. These amino acids, the individual ones, were usually made under very impure conditions and used for animal studies. We actually, between the 3 of us, got agreement with the Pediatric Society. We solved the problem by getting the pure amino acids available, and then worked with the Pediatric Society to have these amino acids distributed by poison control centers, because a physician seeing a child in difficulty having taken a substance they should not have would normally call the poison control center to find out what the antidote was. And so we solved the problem, and I was absolutely delighted.
Great. Okay, so let's move forward, though. Get the bioequivalence, bioavailability. So.
Right. Well, my next assignment was on a drug called phenylbutazolidine. The West Coast firm submitted a new drug application for butazolidine, a generic version of butazolidine, and it was about 65% as bioavailable as the innovator. The FDA had not really any mechanism for receiving this information at that time except through an IND. So this firm, they did not have a generic drugs division at that point. So the information came to me. The physicians really didn't know what to do. They thought that, okay, they had a blood level, so it should be fine. I decided that it should not be, and I rejected it. Marion Finkel went along with that, and the application was rejected. It was not marketed. But I became aware then that there was a unit in the FDA that was looking at this. It had just been started, and it was headed by Trieste Vitti, a PhD and a former professor who was a former associate of Professor John Wagner at the University of Michigan. And he was hired to develop a biopharmaceutics division, although it wasn't called that in those days. And he was promised the opportunity to hire some folks to carry out the review work. But this was the time of the Vietnam War, and resources were very limited. President Johnson needed as much as he could get in the way of financing to fund the war. So wages for the federal employees were frozen. No additional job hires, and as a matter of fact, there was some downsizing. The people who were downsized were assigned to Trieste Vitti's unit. They were pharmacologists. They had no chemistry background, no pharmacy background, and Trieste just was fed up, and he resigned. He went back to academia. I was put in charge with the idea of trying to develop some people, and that was an assignment back in 1972. And at that point, we had a myriad number of problems. I mean, it was like a war zone almost. There were so many issues that were on the table at that time. But I enjoyed the idea of cutting my eye teeth and really trying to solve the problem again.
So since there were all these problems, and you were like running this unit, how did you get the opportunity to come to San Francisco to be able to?
There were a whole series of problems. First of all, any drug marketed prior to 1938 didn't require any clinical studies. And they really did not come into the FDA for review. So the FDA really, the only criteria then for keeping them in the marketplace was the USP specification. As they met USP specifications, it was presumed to be therapeutically available. After 1938, the FDA required toxicity studies. At first, they were very simple, but they got much more complex. They went from acute studies to acute and chronic. But then there was a serious problem with MER-29, which was a drug to prevent the hydroxylation of demonstranol to cholesterol. And a number of the animals were getting sick and being replaced by the investigator without telling the FDA. But there was also a problem with thalidomide, which had caused phocomelia. And Dr. Frances Kelsey, who eventually got the gold star from President Kennedy for her actions in these two items, went in, investigated the firm, and Senator Kefauver had some hearings after this, and now clinical studies were required. The FDA interpreted the new law to mean that every drug that had entered the marketplace after 1930 had to have clinical studies. Well, we didn't know what drugs were effective, which weren't. They contracted with the National Academy of Science, National Research Council. They determined which ones were, which ones weren't. Those that were not were removed from the market. Those that needed additional studies were given time for that. But in every publication as to what drugs were effective, a sentence was put in that they needed bioavailability studies. The APHA and USP and the FDA's Commissioner's Office were all saying that all they had to do was meet USP. There was no bioavailability problem. There are a number of people in academia who were arguing that there was a bioavailability problem. and that's when I became the director. So we had some arguments inside, but fortunately the commissioner's office heard us and let us develop some contracts to do some studies to show they were wrong. We had a contract with John Wagner on digoxin. He analyzed a Fulgara tablet versus a Burroughs Wellcome tablet and found it was 50% bioavailable. At the same time, just before I became director, a group from Columbia University did some studies on the bioavailability of digoxin because in the private hospital practice, they found that many of the patients were controlled with 1/8 of a milligram and 1/4 milligram dose. But the patients in the public hospital required 1/2 a milligram to 3/4 of a milligram, and then every 3 or 4 weeks they would go out of control. They investigated and found that every 3 or 4 weeks the system would buy the next cheapest generic. So they did a bioavailability study and came to the FDA and showed the FDA the data before they published it, which was very considerate of them. The FDA got samples of the tablets and found out that they didn't meet content uniformity, therefore they should not have been in the marketplace. There was no bioavailability problem. When I became the director, I took a look at that data and I was surprised because while there was probably, they were out of specs by maybe 10 or 15%, they were half as bioavailable. So you had really two problems. The FDA had misinterpreted the data. At the same time, this group, just as John Wagner was reporting his data under contract to us, the same group came in and they had this time made sure that that these products had met USP specs and reported that data, and they had not only manufacturer problems, but they had one company that had a lot that was about 10% bioavailable and another that was about 60%. So you had a batch-to-batch problem. This was also a pre-38 drug, so there was no control. It wasn't regulated. And I had by that time also gotten data from triple sulfa applications from generics that had compared their triple sulfa to the Abbott product, and they were all more bioavailable than the Abbott product. I found out that Abbott had done a reformulation, and they followed all the tests and everything that was appropriate in those days, but had not done a bio study, and the sulfadiazine component of triple sulfa was only 50% bioavailable. They reformulated, and that was all right, but I just decided then that we really needed an in vitro system that every manufacturer could look at their product using this, and if there was a change in the in vitro data, then they really needed to go back and take a look at the in vivo data, do another bioavailability study. John Wagner had looked at a procedure that John Poole had developed using a round-bottom, three-necked resin flask, which was a very difficult procedure, and he also used a paddle. The standard procedure at that time was the USP rotating basket, which presented a problem because it was done in a beaker, and the beaker had a hump in the middle, and so depending upon where the tablet laid, you would get varying results. And at that time, they only had like 5 or 6 that were in the compendia. But the USP was very concerned with me in particular because I was really pushing this. And then at that point, when the commissioner's office was made aware of the digoxin bioavailability problem, they appointed me. They wanted to resolve the issue, but this was a pre-38 drug. So they made me the digoxin project officer, and I was to come up with a plan, go through the Bureau of Medicine to the commissioner's office, that the plan would resolve the issue. We had previously lost a commissioner because Park Davis had submitted, or a generic firm had submitted a chloramphenicol application to the FDA, and their application gave 0 blood levels, but the Parke-Davis gave adequate blood levels. The commissioner rejected the generic application. The generic people went to Congress and a few other friends who were powerful on Capitol Hill and showed that their pediatric dosage form gave blood levels, but Parke-Davis pediatric dosage form did not. So Mr. Nixon had now assumed office, and this commissioner, who was a public health service officer, was quote unquote retired. I was aware of all these issues, and I was very concerned, as to how to do this. So I contracted with a laboratory in Maryland, and I wanted 3 non-sequential lots of Burroughs Wellcome to undergo bioavailability testing. I wanted to make sure there was consistency batch to batch, number 1. Number 2, there was not a really good published assay for measuring digoxin. So I contracted with John Wood and Bill Barr at Virginia Commonwealth University to develop a radioimmunoassay, which they did. And I had the FDA inspectors pick up digoxin samples from all over the country, and we had these samples run for dissolution analysis at the National Center for Drug Chemistry in St. Louis, which was part of the Bureau of Medicine. We got results from them that ran from about 10% dissolved in an hour to 95% dissolved in 5 minutes. I now figured I had 2 problems. I had some bad products in the marketplace, and I had a possibility of having a super digoxin that was going to be more bioavailable than Burroughs Wellcome, which dissolved about 50% in an hour. I wrote up a regulation which the commissioner's office signed off on. And the scientific components of that, the legal people really put it all together, but the scientific components really said that every person who wanted to get a new drug application, well, first of all, we reclassified digoxin as a new drug. Major problem. Secondly, anybody who wanted to get digoxin, a generic digoxin into the marketplace, would have to run a bioavailability study versus a particular lot. And I had purchased a large amount of a particular lot of the Burroughs Wellcome product. That way I can make cross-study comparisons. They had to use that in a bioavailability study. And thirdly, they had to have 3 lots of their digoxin undergo dissolution testing that met the dissolution parameters using what then became known as the FDA Paddle method. Well, this was an onerous method in a 3-neck resin flask. The day after this hit when this went out, the FDA is required to publish this as a proposal, get comments, and then publish it in final form. This time, Peter Hudd, who was probably the most brilliant attorney the FDA ever had, insisted that we not only publish it as a proposal, but we would make that effective immediately. That gave us a chance to get rid of the bad products right off the market, just boom. And so as soon as that proposal went out, Burroughs Wellcome was very concerned. They had a product which had been very consistent from the 1930s. It was well controlled. They called Commissioner Schmidt, who was a cardiologist from the University of Illinois, and Commissioner Schmidt stayed the regulation as far as the in vivo requirement was concerned and the reclassification as a new drug. However, the in vitro requirement was still there. And we made it quite clear that anybody who submitted an application to the FDA, the inspectional force would be in their plant to make sure they were making it in proper conditions. Nobody submitted any additional application during the entire time I was with the FDA. But then there was also, we had to do something with the dissolution method. So we assigned the Art Steers, who was the director in St. Louis, to perfect the dissolution system using the FDA Paddle. He took the beaker, cut off the bottom, put in a round bottom, and then we used that with the paddle. There was always the problem on individual analysis, and so they made this with 6 vessels driven by a chain, so all of the data had the same RPMs. And we got some very good data after that. That became known as the FDA Paddle Procedure, but the USP was not happy with us, and there was some real controversy. Senator Kennedy heard about this, and he convened the Office of Technology Assessment, which Congress had passed some years before, but nobody had used. Your professor, Sid Riegelman, was a member of that. Ken Melman, yes, also from the University of California. James DeLuzio, the dean at the University of Texas, was. They had 8 other physicians and a statistician. They came down hard on the FDA because the FDA was saying that there was no bioavailability problem. They determined that there was. They also came down hard on the FDA because they had not put in enough manpower to really do the surveillance that they should have done. But they really came down very hard on the USP, and they made it quite clear that the USP procedures did not assure clinical equivalence, that many of the methods were outdated and they needed to upgrade it. That solved some of the problem. But we still had, the state of Florida had passed a law saying that any product that met USP standards could be marketed in the state of Florida. Many of these were made in New York. The FDA sued in both states and lost in both states. In those days, there was no law establishing an ANDA, or abbreviated new drug program. And some generics filed their abbreviated new drug applications with us, but some took us to court. We went to court in New Jersey, and Dr. Bernie Cabana and Dr. Kraut, who was the head of the Bureau of Medicine at the time, testified for the FDA, and I was the observer sitting in the court trying to make sure that all the testimony that we needed got in. The FDA won that suit. Differences in several circuits went to the Supreme Court, and the Supreme Court ruled in the FDA favor. So we began to put some of these issues to bed.
Yeah. So, Jerry, I'm going to have to stop you. This is a fascinating history, and it doesn't exist anyplace else. It's on this tape. And every pharmacy student learns about these issues, but not to the level. But we're going to run out of time, so okay. I want to get into some other issues that relate to it, because you really had a big impact. Let's talk about your presidency at AAPS. I think that we reviewed ahead of time that distance learning, e-learning was one of your goals that you were trying to push, and let's talk about that and what you think we accomplished.
Okay, when I was in the FDA, we were trying to solve some of the problems, controlled release and other issues. And the idea was to bring academia and industry, or FDA and industry together under an academic overview. But this was very expensive for the industry to get their people to a workshop all the time. So when I became president, there were 2 things I really wanted to do. One was to establish a strong distance learning program. And we had Andy here who really was very capable, but we weren't putting any resources into it. We finally got that up off the ground. We started that during my term, and I'm very, very proud of that. The other thing I really wanted to do was the outreach program with Stacy May, who was reaching out to all the schools of pharmacy. And as you know, at one point I contracted with you to have all your pharmacy students look at certain drugs to see if indeed there were assays to do bioavailability studies, because the FDA didn't have that enough people to go ahead and do that. So these were two very important things, and I think, given the number of student chapters we have and the number of foreign student chapters and what they've done with what they now call e-learning, I'm very, very happy that we got that done.
Good. Congratulations. I think that was a very important steps for the association. Let's talk a little bit about the future. Do you think, in essence, we've solved most of the bioequivalence, bioavailability problems?
No. I went to an IBIBC meeting here, a symposium here, and we must have had 250, 300 people in there, and it was interesting because they were looking at the BCS. Permeability only became an issue after our first controlled release workshop, and we were trying to figure out how we could get dissolution, meaningful dissolution for controlled release products, and that was the first time it came up. Gordon Amadon came into the agency just as I was leaving, and he and Vimal Shah came up with the biopharmaceutical classification system, and I read your more recent publications having to do with transporters. And so they were very worried about why they could not get an IV, IVC with class 2 drugs. So I pointed out to them that there was a transporter issue, and there was, if you had lack of saturation, you may not be able to. So some of these people had not read that. But anyway, I see this progress, this tremendous progress from this little rotating basket in a beaker to where we are today, and I know we have a lot more evolution to go through before we're there, and some of the work that you're doing is going to determine that.
Thank you, Jerry. So let's talk about AAPS. You must be very proud, like we all are. Where do you think we're going in that?
I am just stunned. I remember the first meeting we had here in Washington in 1986 at the Hilton Hotel. And I thought that was wonderful. I didn't pay my dues real quick, so I was this 2,000th member or something, but I had been a member of the Academy of Pharmaceutical Sciences, and I was fully supportive of what you were trying to do. And I was really made an effort when I was in the FDA to have as much involvement of the FDA with this, because I saw this as mutually beneficial. And fortunately, AAPS really worked with the FDA on a number of issues. You and Jerry Goyen were on a number of panels in the Commissioner's office. We've made tremendous progress, and I think it's really facilitated good regulation. And in addition, it's gotten the word out. What I'm really stunned by is that at that time, 80% of the members were from big pharmaceutical companies. Now it's only 20%. And our foreign component has grown tremendously. So the impact that this society is having worldwide is wonderful. And it's also beginning to think in terms of working with the many small groups that we've not thought of in the past. And so we have some work to do on that area.
Okay. So Jerry, thank you very much for sharing your thoughts and history with our audience, and we greatly appreciate your willingness to come today.
Thank you, Les. Thank you very much.